So we'll start with cholesterol synthesis today. It sort of picks up where we left off last time with doing the ketone bodies before we did fatty acid synthesis. They have a lot in common, but they differ a little bit. But before we get into the actual chemistry of synthesis, I want to let you notice some other things that go along with cholesterol. The picture on the top right is the heart disease rate in the U.S. from 2002 to 2007, listing adults age 35 plus that died annually from heart disease, whatever the cause of the heart disease might have been. There's a few causes, but they pulled them together to make this chart. And at the time when I did this lecture back then, it seemed like a lot. And you see it's kind of focused around the South, like all of Mississippi. So they redid this study. They do it all the time, every year. And I grabbed a new chart, the one below, for 2014 to 2016. I grabbed that one a couple years ago. And at some point I'll grab a new one. But what stands out is not that it's the same pattern, where it's focused mainly the South, but the numbers. If you look at the chart at the top, the states with the areas, counties actually is what they were doing it by, with the highest rates were in the 112 to 254 range per 100,000 deaths, or per 100,000 people. That is the lowest possible category in the graph at the bottom. Everything is much higher than that by 2004 to 2006. So that's what should stand out about these two graphs, that it's not that the problem is still there, but it's gotten much worse. So, and it still follows the same exact pattern of which counties are in trouble. So the lowest rates today are the highest on the graph from 2002 to 2007. And I'm sure another one of these will come out in a few years and we'll be appalled at how much greater it's gotten since then. Right? So we definitely have to fix this. So what's causing these problems? Well, for most of the cardiovascular disease, it's caused by the diet. Right? We have a high-fat diet, which is entirely too much. Okay? So one of the main causes is what we eat. Another cause is genetic, of course, and we have certain illnesses that are in that category as well, but it comes a lot from our diet. So let's see what we do with, or how we make cholesterol, since you don't have to eat it. You get all the cholesterol you need, assuming all your enzymes work, by making it yourself. You do not need to consume cholesterol. Okay? So how do we make cholesterol? It starts out a lot like our ketone body synthesis. In fact, it's exactly like it in the beginning. We're going to make it entirely from acetyl-CoA. Yes, why do we need to make cholesterol? Well, we use it for lots of things. It's put in the membranes, and I'm sure you guys covered this last semester. When you put cholesterol in a membrane, it modulates the fluidity of that membrane. By having a cholesterol molecule there, and you remember the shape of it, it's a big fused ring structure. If you have a amount of cholesterol in the membrane and the temperature of the membrane rises, the phospholipids tend to start to fly apart, right? And the membrane would completely disrupt at a high enough temperature. But with the cholesterol there being a very rigid body, it doesn't flex. It gives those phospholipid tails something to hold on to. Conversely, on the other end, if you have a purely lipid membrane, or purely purely phospholipid membrane with all the fatty acid tails, and you cool it down, they tend to form a solid, which would also be non-compatible with cells living. So cholesterol being there, not because it's rigid this time, because it's a bulky molecule, those molecules of tails, of fatty acid tails, can't form a solid. They have to go around this bulky cholesterol. So it keeps the membrane more fluid at a higher and a lower temperature than without it present. So it allows the cells to survive a wider range of temperatures. And we do this in mammals in particular. We do it in some plants, and we also do it in fungi. And we'll talk about the differences in those later. We also use cholesterol to make other things, like our steroid hormones, progesterone, testosterone, estradiol, cortisol, and so forth. And it's also a precursor for vitamin D, right? So we use cholesterol to make vitamin D, right? As opposed to the myth that vitamin D comes from the sun, which is not true. Vitamin D, the molecule, does not fly through space to get to Earth, to get to humans. The ultraviolet light from the sun converts a molecule of cholesterol into vitamin D. That's what it's used for. Okay, so back to how we make it in the beginning. So we're going to start with molecules of acetyl-CoA. So in stage 1, I'm calling it here, we're going to convert 3 molecules of acetyl-CoA into a molecule called mevalonate, Right? Mevalonate has 6 carbons. They come from the 6 carbons of the acetate groups on the 3 acetyl-CoAs. We did this exact set of reactions when we did ketone body synthesis. Okay, up to that point, it's identical. Okay? We're going to, well, not mevalonate, but up to HMG-CoA, it's identical. We're going to take the mevalonates and turn each one into an isopentenyl pyrophosphate, which only has 5 carbons. You could probably guess where the other carbon is leaving as. Right? This occurs in the cytoplasm. So why are we doing this here? Well, it must make sense that we do it in a place where the molecules are soluble. So acetyl-CoA, mevalonate, isopentenyl pyrophosphate, the HMG-CoA we're making, all are water-soluble molecules. So we're going to do this in the cytoplasm. In the next part, we're going to take 6 of those 5 carbon molecules, the isopentenyl pyrophosphates, and combine them into squalene. So how many carbons would squalene have? Taking 6 5-carbon molecules, I'm not losing any carbons, and I'm making squalene. So squalene would have 30 carbons. Why does this happen in the ER, in the membrane? Well, at that point, these molecules have no other groups on them but hydrogen and carbon. Right? That's it. They're pure hydrocarbons. We've lost all the oxygens, we've lost all the phosphates. So this thing is entirely insoluble in water. So it needs to be in a lipid environment. So it's going to be in the membrane. And then finally we take squalene, which has 30 carbons, and cyclize it. We're going to make those 4 ring structures you saw in the cholesterol molecule from squalene. We'll lose a few carbon atoms along the way as methyl groups. So we'll go from 30 carbons down to 27. This also occurs in the ER because, again, not soluble in water. So back to the beginning. Let's talk about how we make that mevalonate molecule from acetyl-CoA. Okay, so we're going to start off with acetyl-CoA, shown on the left there. Right, I'm going to take a second acetyl-CoA molecule and make the 4-carbon molecule attached to CoA. Right, this is exactly just like the ketone body synthesis we did. We went over that reaction already of what attacks what, and I'll show it again in a second. We take a third acetyl-CoA molecule, add it on. Now we have 6 carbons attached. Remember, because it also attacks the same carbonyl, it will be necessarily branched. So our HMG-CoA is branched. It has 5 carbons in a row and a methyl group. It also has an OH group, which came from that 3rd carbonyl that we allowed to stay as an O- and then protonate with the water molecule. So up to this point, it is identical to ketone body synthesis. We make HMG-CoA. So with ketone body synthesis, at this point, we lost the original acetyl-CoA and we go down to 4 carbons, right, and reform the 4-carbon molecule. Of course, we then would reduce it, or it would spontaneously decarboxylate and give us acetone. Okay, so we're not going to let that happen here. We're going to take HMG-CoA and immediately reduce it. Okay, so we're going to take off the CoA at the end. So we're going to cleave off the CoA. If you cleave that off using water, essentially, it becomes a carboxylic acid. Right? So it'd be a 5-carbon carboxylic acid on the end, on both ends, in fact. We're also going to take that carboxylic acid and reduce it twice, from a carboxyl group to an aldehyde to a primary alcohol. So the molecule on the far right there, you see that carbon on the right, the 5th carbon, is now reduced completely to a primary alcohol. Right? That's called mevalonate. Right? There's no CoA left. It does have a carboxyl group, and it has 2 hydroxy groups. Right? So it's still got a 3-hydroxy and a 3-methyl, but it has no longer a CoA on the end. Right? So it's just mevalonate. Right? In the next steps, we're going to take mevalonate, phosphorylate it 3 times with 3 ATP donating the phosphates. Right? We'll talk about where those phosphates are going to go in a second. You probably could guess they're going to go on the hydroxy groups. And then we'll eventually lose a carboxyl group. You probably can guess which one that is. And we're going to lose 1 of the phosphates, leaving 2 behind. And that gives us the 5-carbon molecule, the isopentenyl pyrophosphate, okay, which has the form of an isoprene unit. And if you remember from, maybe you did this in organic lab or you studied this in biology, isoprene units are the basic building blocks for long chain molecules in plants, right? They make these long polymers in plants. So we're going to do the same thing here, just not so long as a few hundred copies. It's only going to be 3 copies. Right? Okay, let's look in detail at that first part, where we turn 3 acetyl-CoAs into our HMG-CoA and then to mevalonate. And so on the left here is your first 2 acetyl-CoAs. They condense and make the aceto-acetyl-CoA, the 4-carbon molecule. Right? We lose the CoA by a thiolase, And now we have a very unstable beta-keto arrangement. Okay? We don't want to keep it that way. We take yet another acetyl-CoA. It attacks the very same carbonyl and forms the branched HMG-CoA. Okay? Pay attention to the name of the enzyme here. It says HMG-CoA synthase because I'm synthesizing a CoA. Everything I've done up to this point is fully reversible. I could turn HMG-CoA back into acetyl-CoA, and of course back into two individual acetyl-CoAs. Everything up to this point, fully reversible. I haven't committed to this yet. At this point, if I take HMG-CoA and do the next step, it's an irreversible reaction, and I'm committed to making cholesterol. So much like we did in fatty acid synthesis, when you turn acetyl-CoA into the 3-carbon carboxylated version of it, which was malonyl-CoA, right? That was the committed step, irreversible, you're going to make a fatty acid. Here's our committed step here, right? HMG-CoA reductase. Once I turn HMG-CoA into mevalonate, there's no going back. So it might be a good idea to regulate it here. Okay, so to turn HMG-CoA into mevalonate, we're going to do a couple things. We're going to take the carboxyl group at the very bottom, which is attached to CoA, cut off the CoA, like we said, exposing a carboxylic acid or carboxylate, and then reduce it twice. Our cofactor for that, since we're doing a synthesis pathway or an anabolic pathway, is going to be NADPH. So 2 NADPHs are consumed to make NADP+, and I convert carbon number 5 there from a carboxyl group to its primary alcohol. Right, same molecule drawn at the bottom is also at the top up there. It's just turned on its side so you can see the carbons labeled 1 through 5. It's still mevalonate. And then I said I'm going to phosphorylate it 3 times. So the first place I'm going to phosphorylate it is on number 5, as you expected. Put the phosphate on. That's the farthest from the other negative charge. That makes sense. In the next reaction, I'm going to phosphorylate the same phosphate again, making a pyrophosphate. And in the third reaction, I'll phosphorylate the other hydroxy group, right? The one on number 3. Right? So I have phosphor groups on 5, 5 again, and then back on number 3. At this point, the carboxyl group on number 1, or the carboxyl group that is number 1, can leave as CO2. When it decarboxylates, it leaves behind a lone pair. That lone pair will form a bond between the carbon number 2 and carbon number 3. And of course, carbon number 3 can't have 5 bonds, so we expel the phosphate group. Essentially, we use this phosphate to remove the OH. We've seen that game before when we talked about the nucleotide synthesis. So a loss of CO2, irreversible, and a loss of phosphate, great leaving group, makes this reaction go forward. So I put 3 phosphates on at the expense of ATPs, lose a CO2, lose a phosphate, and I'm left with a 5-carbon molecule. It's still branched. It's 4 in a row with a methyl group, and it has a pyrophosphate still on it. That's still an excellent leaving group we're going to take care of in a second, or make use of in a second. What I want you to see at the bottom of the screen is that isopentenyl pyrophosphate, you see the double bond that formed between its carbons 1 and 2, right, could also migrate between 2 and 3 now. Right? What is carbon 2 and 3 now? So that double bond can be in either place. We have 2 forms here. We have the isopentenyl, and then we have the dimethyl allyl. Right? We're going to see we need one or the other to do the next reaction. We need one in one form and one in the other, of course. Okay, so to summarize this slide, you're going to take 3 acetyl-CoAs, form your HMG-CoA, just like we did with ketone body synthesis. But here's where things differ. We're not going to lose one of those. We're going to reduce it and cut off the CoA to make mevalonate. From there, we phosphorylate it 3 times, decarboxylate, lose a phosphate, and I have my 5-carbon isopentenyl pyrophosphate, which can exist in 2 forms. Double bond between the first 2 carbons or between the second, third carbon. Okay, the other thing on this slide, please don't get confused, is when you make HMG-CoA, it's HMG-CoA synthase. That should make sense. We're synthesizing it. That is not the controlled reaction. That is not the committed step. That is not the regulated reaction. That one's fully reversible. The committed step is the next one, is HMG-CoA reductase. Don't confuse synthase with reductase. Okay, so at the next step, I have an isopentenyl pyrophosphate, right? So it can exist in 2 forms. We're going to take 6 of these together and make squalene at the bottom. So what would drive this reaction forward? How can I take those molecules and put them together? I mean, making a carbon-carbon bond is not easy, so I need some way to make this more of a favorable synthesis. Anybody have a guess? No? I mentioned it earlier. We still have these pyrophosphates on the end, right? And we're going to take those as good leaving groups. They're going to leave as pyrophosphate, which of course can get subsequently degraded to orthophosphates. So we're going to take the 5 carbon molecules and combine them together to make a 10 and then 15 carbon molecule. Then we're going to take 2 of those 15 carbon molecules and put them together and make 30. And so it happens in 2 parts. In the first part, on the left there, we have the 2 5 carbon molecules with their pyrophosphates. The one on the far left is in the dimethyl allyl form, where the double bonds between 2 and 3. And the one on the right, that's added to it, has the double bond between 1 and 2. Okay, so if we take those together, you can see that the one on the right, the isopentenyl, can make a bond to the first carbon of the other one, or the last carbon of the other one from our point of view, and the pyrophosphates could be a leaving group. Okay, so I attach one carbon chain to the next by a single bond. Right, so I have to, you can't see my mouse, I know, but you will in the recording. I have this carbon here on the dimethyl allyl lose the bonds to the pyrophosphate, and it makes a new bond to this carbon. And when it does so, when this one makes a new bond to here, this double bond has to migrate to the position between 2 and 3. So it looks a lot like the dimethyl allyl when we're done with that reaction. That would be right here. Okay, so putting those together, I have a 5 carbon pyrophosphate and another 5 carbon pyrophosphate. Put them together, we lose the pyrophosphate on one of them. It makes a geranyl pyrophosphate. And geranyl has 10 carbons. We do the same reaction again. In this case, the bond here, right? Or so the carbon here, this bond to the pyrophosphate is lost. It makes a new connection to the next carbon of the third isopentenyl pyrophosphate I'm using. Again, migrating the double bond over to the left, to the right to make farnesyl pyrophosphate, which has 15 carbons. So this is a head-to-tail assembly. I'm taking the last carbon of one and attaching it to the first carbon of the next at the loss of a pyrophosphate. So I do that twice and end up with a 15 carbon chain. It's got 3 methyl groups, so it's only 12 carbons long, but it's got 15 carbons, right? And I take head-to-tail assembly to make those. I do those to make two 15 carbon farnesyl pyrophosphates. And on the right, I'll connect them head-to-head now. Instead of putting it head-to-tail and making this longer, we're going to take 2 of these and put them head-to-head and lose both pyrophosphates. At the same time, I need to get rid of the extra double bond that would create. Right? I have an extra double bond between them. So I'd reduce that with NADPH one more time to get a single bond. So this is a head-to-head assembly, and this one's squalene synthase. Right? It's the very last step. So one enzyme, prenyl transferase, does all the steps on the left, assembling 5 plus 5 to make geranyl 10, 10 plus 5 to make farnesyl 15. Same enzyme did both. And then I have a different enzyme, squalene synthase, to take the 2 head-to-head to make squalene. Okay? And you see what's left has absolutely nothing but hydrogen and carbon in it. It is not soluble in water at all. So in the assembly of this, we start in the cytoplasm and we end up in the membrane. So these enzymes must put that product in the membrane, the squalene. And from there, what happens to the squalene is we have to cyclize it. I redrew the squalene here a little bit differently so you can see what rings are going to form from which parts. Unfortunately, we don't number them that way. You learned last time, I think, the numbering system for cholesterol. Right? So we go from 30 carbons down to 27. We lose 3 methyl groups, one at number 14. That's the blue arrow. It's not there anymore. And we lost 2 from the position at number 4, which were originally on the first carbon, or second carbon over here. We lose both of these methyl groups and one of these. Right? So how does that happen? All right, so I drew it several ways here. We're going to do it for the way the plants do this. They make their sigmasterol, and humans and other animals do this. We make cholesterol. And then the fungi also do it. They make something called ergosterol. Okay, and we'll talk about the differences in these in just a second. Well, start at the top with squalene, right? And we do that very difficult reaction. It's not so easy to do. It's called a monooxygenase reaction. So between carbons 2 and 3 of this chain, which will no longer be 2 and 3 once we make cholesterol, but 2 and 3 of our current squalene chain, we have a double bond. And we're going to use O2 to put a oxirane ring between carbons 2 and 3. So it's a 3-membered ring, which is not stable. We've seen this with other things like our signaling molecules that we made from lipids, our progesterones and leukotrienes and things like that. But we're going to put a double bond into a oxirane ring, right, which then immediately will break, of course, and we end up with a hydroxide on number 3. That's what we intend to do all along. Right? And so we put these things together and make this squalene 2,3 epoxide, and it can be turned into a regular old OH in virtually the same way for all these reactions. What differs in the way plants do it versus fungi versus animals is what we do at the other end of the molecule, right down there at number 17, right? What will be number 17. So in plants, the modifications are quite different down there. So we have this longer chain with more branched. In ergosterol, we don't have much of a branching, just a few methyl groups and a double bond in it. And in mammals, or in other animals, we end up with a long chain down there that's branched at the end, an iso group, and no double bonds. So the main difference is down there at the end. The rest of the molecules look almost identical. Okay, so if you could imagine coming up with a drug that would inhibit the reactions that only fungi use, it wouldn't be harmful to plants, it would be harmful to mammals or animals. So it could be a good antifungal drug if you inhibit the production of ergosterol. You could do the same with plants. You could come up with a drug that would inhibit the enzymes that are unique to plants in that case. You can get some herbicides made that way, and they wouldn't be harmful to animals or fungi. You could also come up with ones that work against the animal versions of those enzymes. And we actually have one of those. It's not so deadly, but it does inhibit cholesterol production, and those are our sterols. Or not steroids, but statins. Right? We're going to talk about those too. So if you can find a unique enzyme that's different from the other two in this split pathway, you can make a drug that's unique to that system. And it probably will not harm the others. Right? But if you go back and inhibit the squalene monooxygenase, you're going to inhibit all 3 branches of the tree there. Okay? Do I need you to memorize these reactions and what these structures look like? No. But I need you to know that there are differences between the 3. Right? So know that the plants make it differently from the fungi, differently from the animals. Okay. So looking at the human, or the version of this, the animal tree of this, let's see where this happens and how we can stop it. Because we get enough cholesterol in our diet, right? You don't need any in your diet, in fact, but we get so much in our diet that we shouldn't be making very much at all. So how do we stop this enzyme? Which enzyme am I talking about? Not the ones on this page. Go all the way back to the HMG-CoA reductase, the committed step in the pathway. So we're going to make an enzyme that inhibits that. If you could get plants or fungi to actually take this enzyme, which doesn't make any sense, but they would also not make their version of cholesterol. Okay, so how does this thing work? How do we inhibit this enzyme? Well, the plant or the animal or the fungi do it already a fairly good job on their own. So the first thing, you probably talked about this last semester when you were doing regulation in your DNA or RNA lectures. So you had a sterol regulatory element, right, which was a protein that was found inside the cell. And when sterols, like cholesterol, get to the cell, it's not difficult to go right through the membrane because they're lipids. But they can't really get to the nucleus, right? From the sterols or cholesterol's point of view, the nucleus is separated from it by an ocean of cytoplasm, right? It can't cross it. So it uses this little protein to carry it there. So the protein found on the inner leaflet of the cell membrane binds to the cholesterol, picks it up, travels all the way into the nucleus, and then goes and acts as a transcription factor, right? Shutting off the production of HMG-CoA reductase. So this is control at the transcriptional level. So this regulatory element binding cholesterol prevents the production of HMG-CoA reductase mRNA, right? We won't even make the message. So it's the earliest levels of control. We don't want to make this enzyme. Next, some of that mRNA, in fact, does get made. It's not turned off completely. It's drastically reduced, but it's not turned off completely. And if there's still cholesterol around, it can bind to the mRNA, or bind to a protein that binds to the mRNA, and stop it from being translated. The ribosome will not translate that mRNA if that's bound to it. So now we have translational control. But a few of them will get made, right? Again, drastically reduced at this point. But a few of them will get translated into the HMG-CoA protein, HMG-CoA reductase protein. So what happens next, if there's still cholesterol around, we can have the cholesterol activate a kinase, and that kinase phosphorylates HMG-CoA reductase. And upon phosphorylation, it's turned off or turned down. It doesn't work as well. So we have a third level of control. This is a post-translational allosteric control. So 3 levels so far of not making this thing, or not making it work. And then on the 4th level, what the cell does is if it finds any HMG-CoA reductase in excess, a protease, again activated by cholesterol, destroys the enzyme. So it really doesn't want to make this stuff. If there's any cholesterol around, let's not make it. Why such a harsh regulation? Because it's expensive to make it. If you look back, we spent 3 acetyl-CoAs, each worth the equivalent of 10 ATP in energy, right? And then we spent 3 ATP for each of those 6 molecules that we turned into squalene. So this is quite expensive, right? So 3 acetyl-CoAs to make each mevalonate, and we're going to make 6 of those things. So that's quite a few acetyl-CoAs. And then 18 ATP to make that into squalene. So this is not something you want to do and waste all your ATP when you don't need cholesterol. Finally, humans have come up with a way to do it. On top of these 4 things, we've initiated another control mechanism, and we've made an inhibitor of the enzyme, right? A competitive inhibitor, these are your statin drugs, that inhibit HMG-CoA reductase. Right, so in addition to the 4 things here, I guess its position at number 5 is not the best. It should be between number 3 and 4, I guess, because it's like number 3, it just turns off the enzyme, but it doesn't degrade it. So, but it's in addition to the cell's normal 4 ways of preventing cholesterol synthesis. So we've added a 5th. Okay. Right, any questions on the cholesterol real quick? How it's made, how it's regulated, before we move on to the other proteins? Okay, so the other proteins I'd like to go over, besides how we make cholesterol and all of its associated synthesis enzymes, are how do we get these things moving around through the body, right? Because cholesterol and other lipids like triacylglycerols aren't very soluble in water, so they cannot travel through the bloodstream on their own. They need some kind of carrier, a taxi. So we're going to go over 4 of the taxis that are used. So I highlighted them at the top of the screen here. There is a 5th one in the middle there that I've blocked out, and it's just a precursor to the other 2 below it. So not so much of a additional feature, just a precursor. So I want to talk about them in 2 groups. The top group is called chylomicrons and VLDLs, very low density lipoproteins. We'll abbreviate it as VLDL. Okay? Both of those have a low density compared to the other 2, right? The one's termed low density and high density lipoproteins, but they're all very, very close. They're all around the density of water, which makes sense. But these are slightly less dense than the others, that's why they got their names. And then the other 2 at the bottom are called low density lipoproteins and high density lipoproteins. That doesn't mean they're very high density, but compared to the others, this is the order of density increase. And you can see the relationship in their density by looking at their size. Right? They both carry lipids. They're made of protein and they have lipids in them. But as you go from top to bottom, the proportion that is protein increases and the proportion that is lipid decreases. That's why it gets more dense, because protein is more dense than lipids. So at the top, you look at the chylomicrons, they're 75 to 1200 nanometers in diameter. That's huge. Right? There's a picture of it on the bottom right of the screen. Chylomicrons in the upper left of that 4-picture cluster, and it's only shown at 60,000 times magnification. The other 3 photos are at 3 times that magnification. So to put to scale, I couldn't find one to scale, but to put it to scale, make the picture with the chylomicrons 3 times bigger. Then it would be to scale with the others. Right, so yes, it's the largest of the group. The VLDLs are 30 to 80 nanometers in diameter, so that's shown on the bottom left picture of the cluster, and you see they're much bigger than the other 2 on the right, but clearly smaller than the chylomicrons, even not to the scale. Right? Now, underneath that, we have low-density lipoproteins shown in the upper right of the cluster of pictures. Right? They look like little viral particles over there. Right? So LDL shown at 180,000 times magnification, clearly smaller than the VLDLs, but much bigger than the HDLs. And finally, HDLs, which are less than 20 nanometers in diameter, you see they're very small over there. So they're more dense. They contain numerous different proteins, and this is what a lot of these cholesterol tests test for, is the concentration of these proteins in the blood, not necessarily the cholesterol itself. And their physiological roles is what I want you to know. So looking at the top pair, the chylomicrons and the VLDLs, their composition has very little protein, that's the P on the right. It's only 2 or 8% of the molecule, or the cluster, and it contains mostly triacylglycerols, right? 86% and 52%. By far, the majority of its cargo is triacylglycerols. They do carry a few other things, but that's the main cargo, right? And they have very little protein to it, so these are very lipid dense, not such bad choice of words. They're lipid dominant molecules, right? They're not very dense at all. The other 2, the LDLs and HDLs, have much more concentration of protein, 21 and up to 50%, right? So it's much more protein than it is lipid, and they carry mostly cholesterol and cholesterol esters. That's what I boxed in red for you. Yes, they do carry some phospholipids and they carry a few triacylglycerols, but predominantly they carry cholesterol. And that's what I want you to know. The chylomicrons and the VLDLs carry triacylglycerols, and HDLs and LDLs carry mostly cholesterol. Now, the only other thing you need to know is not so much what they're carrying, but where is it going? From where do I get this cargo and where do I drop it off? So for the chylomicrons, they pick up their cargo of triacylglycerols in the backside of intestinal cells, in the capillaries of intestinal cells. So whatever food you eat goes through the small intestine, goes into the intestinal capillaries, and it ends up in the bloodstream there. The chylomicrons pick up these triacylglycerols, because again, they're not soluble in water, I need a taxi, and from there it goes through the hepatic portal system, through the veins to the liver. Right, chylomicrons, that's the end of their job. They've done their duty. That's the only place they take their cargo. So they pick them up at the small intestines, drop it off at the liver. Their goal now is to get back to the small intestine and pick up more. But you can't really go backwards through the bloodstream. It doesn't allow that. You also don't want to randomly flow through the bloodstream and hope you end up back at the small intestine capillaries. It's very unlikely. So they find another way back. The chylomicrons hop in the lymphatic system from there, from the liver, get in the lymphatic system, and make their way back to those small intestine capillaries. So it's like Atlanta. You can't go backwards on I-285. You'll just die, right? You have to find a surface street to get back to where you want to go. You also don't want to make the loop around the entire city, which would take forever, and you're not going to end up at the right exit, right? So it's kind of an analogy for that. Let's find a back street to get back to the small intestines. For the very low-density lipoproteins, the VLDLs, they pick up where the chylomicrons left off. They pick up those triacylglycerols from the liver and transport them out to the rest of the body, mostly to other fat cells. Right? So they bring out the triacylglycerols from the liver to the rest of the body. So it's kind of a relay race between the two. Chylomicrons dropped it off at the liver. These pick it up where they were left off. So they hand the baton on. Okay? And then, of course, they transport it out, never back to the liver. In the LDL and HDL case, they transport it in opposite directions. It's not a relay here. It's completely opposite directions. The LDLs take the cholesterol that was made in the liver, like we just talked about its entire synthesis, mostly done in the liver, stored in the gallbladder. It picks it up from there and transports it out to the rest of the body, again, mostly to adipose tissue. But every cell needs it, so it transports it out. The opposite takes place with HDLs. They pick up excess cholesterol from all around the body and bring it back to the liver for storage. Okay? So if you're eating a lot of cholesterol in your diet, of course it goes in through the mouth, ends up in the stomach, in the small intestine, and in those intestinal lining cells. LDLs coming from the liver normally transported out to the rest of the body. But if they're picking it up from the small intestinal cells and bringing it back to the liver, it still has to transport it out to the rest of the body from there. Remember, all the blood that comes from those intestines goes through the liver. It has to go through customs first. So if you're eating a lot of cholesterol, the number of LDLs you need is going to be higher because there's more things to transport. The number of HDLs will also be higher, but not in the same proportion. You need a lot more transporting to fat cells than from fat cells. So in a normal, healthy individual who's not having the high-fat diet of typical Americans, you're going to have about a 3.5 to 1 ratio of LDLs to HDLs. There's always more LDL than HDL. Like any good company, you ship more products out than they are returned. Okay, so 3.5 to 1 is great. But if you have an excess cholesterol that you need to store in fat cells all over the body, the number of LDLs is going to increase at a faster rate than HDLs. So those with high cholesterol, they measure this LDL to HDL ratio. They call it good and bad cholesterol. You're actually measuring the protein concentrations, not the cholesterol. But the LDL to HDL ratio might be 5 to 1, 10 to 1, 100 to 1, which is very bad. Okay, so to summarize this slide, you need to know triacylglycerols are carried by the chylomicrons and VLDLs in a relay race, and which direction they're going. And the cholesterol is carried by the LDLs and HDLs in opposite directions. Okay, and here's an example of what could happen if there's too much cholesterol around and too many LDLs carrying it. LDLs are very bad carriers. They're very bad taxis, right? It's more of a rickshaw than a limo, right? So if you can imagine piling 60 people in a rickshaw, everybody knows what a rickshaw is, right? It's a cart pulled by a human instead of an animal, right? And people ride in the back of it. So if you can imagine you piling 60 people on one of those things, being the cholesterol packed into an LDL carrier, it doesn't have a lot of protein. There aren't seat belts, there aren't windows, there's nothing to hold you in. Somebody's going to fall out. And if you fall out on the street and you're a cholesterol molecule in the blood, you're basically roadkill. Right? So you're going to get run over and it forms a plaque. It's no reason it could go in solution again. Right? There's no street sweepers like LDLs. So eventually it'll build up, and if you look at the picture up there, the A represents what was, it's a slice on a slide now, but it was an open artery where all that middle area was patent to let blood go through. And on the right, you see it's mostly occluded. Maybe quarter to a third of the opening is left for blood to flow through. Right? And this is most likely a coronary artery because that's where this tends to fall out. If you're traveling on a street in this overcrowded rickshaw, the most likely time for you to fall out is when it makes a sharp sudden turn at high speed. And that's where the blood's exiting the aorta and going into the coronary arteries. Right? So they're most likely to fall out there. And they get deposited onto the inner surface of the artery. An example of one of those statin type drugs at the bottom is that inhibits competitively HMG-CoA reductase. It looks a lot like cholesterol, right? It doesn't look exactly like cholesterol, but it looks a lot like it, right? It has the ring structures and so forth. And all these companies, drug companies, are doing is they change the foliage on this molecule. All these methyl groups you see and some of the OHs, and they extend it, throw some other flowery chemicals on there. It might be a nitro group, or put an amino group here, or change this to a different position for the carbonyl, or whatever it might be. They keep changing those, and they give it a different statin name, right? And it may have a different efficacy. It might last longer. It might be a better inhibitor, whatever. But that's all they do is keep changing the foliage on these things, and it makes another statin drug. But they all generally work the same way. They competitively inhibit HMG-CoA reductase. Right? And then, so the last thing I want to talk about is what we use cholesterol for. Right? So we made it, we transport it around. What do we use it for? We make it into other things. So the thing at the bottom, I know it looks like chocolate, but that does not say chocolate. However, it will help you digest chocolate. Right? So it's called glycocolate, and it's made from cholesterol. Hopefully you can spot the cholesterol steroid core there. We've done some modifications to it. Right? There's a few extra OHs that weren't there before. And the very end of the molecule, or the tail end of the molecule, has been modified. So we've lost a lot of the carbons on the tail, right? And we turned it into a carboxylic acid. That's why it's called cholate at this point. It was cholic acid, and the conjugate base would be cholate. We've also put something on that carboxylic acid in blue, right? The thing in blue is simply glycine, right? So we've attached a glycine, amino acid, to the end of a cholic acid made from cholesterol. So we name it glycocolate. So what's the benefit of this molecule? Well, it's still not soluble in water, right? All those carbons in those rings. But it's a little more water-friendly than cholesterol was. It's got some parts, all those OHs, and it's got a glycine at the end of it, which is very hydrophilic. So parts of this molecule are hydrophilic, parts of it are clearly hydrophobic. It has some character of both. This is your typical emulsifying agent, right? Or detergent. Right? So this is what it's used for. This is an emulsifying agent when you eat fats or lipids. This is released from the gallbladder along with all the other digestive enzymes that are coming through the bile duct. And it helps you emulsify the fats you eat, which essentially increases the surface area that the enzymes have exposure to this thing. Right? The other things we can make from cholesterol are our steroid hormones. So these are the three of them on the right, testosterone, estradiol, and progesterone. And then there's a synthetic version of progesterone called erythrodrone at the bottom. And you see how it mimics progesterone. Right? It doesn't exactly look like progesterone, but it has some of the same qualities. Right? The triple bond in there clearly is not going to function the same as a carbonyl, but it mimics it. So it tricks the body into thinking progesterone is being produced. So this is a synthetic progestin that's used as a contraceptive. And if you take it, the body thinks there's progesterone being produced. Progesterone is generally produced during gestation. That's why it's called progesterone. It promotes or continues promoting gestation. So the body thinks it's pregnant. So in that case, the ovaries don't develop any new ova. That's the whole idea. Okay? Do I want you to memorize these structures? No, but it's pretty easy to figure out which one's which by looking at the foliage on the molecules again. So looking at estradiol in the center, it has two OH groups. Obviously, it's a diol. They're only important carbons 3 and 17. Testosterone, obviously, is a ketone at number 3. The other end is still a hydroxy. Progesterone has two ketones, right? The one on number 3, and then again up there on number 18. Right? They also have differing number of carbons, as you notice throughout here. Right? So we have to look at these and figure them out. Clearly, you can make them from cholesterol. You see the core structure. But if you make them, you can only lose carbons. You can't gain new carbons. So if you put it in order in which they're made, which is this final slide here, and I number them for you at the top. So the top left is regular old cholesterol again. You've gotten used to drawing it. At least you know how to draw the first 17 carbons. And then it has that long tail. Well, the first step we do in this process is to cleave off the tail. Right? Everything beyond carbon number 21 is cleaved off. So it has 27 carbons. Cleave off the tail. The only things left on there are carbons 20 and 21. We put a carbonyl there. And now we have our 21 carbon. Those are all the progestins. Right? So everything here has 21 carbons in yellow. That makes progesterone. It makes pregnenolone and so forth. Right? Everything here has 21 carbons. We can do several reactions to this thing. And the green bars and pink bars and everything else and white bar all represent enzymes that are taking place. So we can turn our progesterone into a lot of our mineralocorticoids, which also have 21 carbons. We haven't lost any carbons. We're just adding OH groups or oxidizing and reducing things. That's all these things are. They're a bunch of hydroxylases. So all our mineralocorticoids, 21 carbons, are made from the progestins, which also have 21 carbons. If I take the 21 carbon progestins and lose 2 carbons, switch to the tail again, number 20 and 21, and leave only the core, 17 carbons plus the 2 methyl groups, I'm down to an androgen, and that's shown in blue down here. So our androgens, such as testosterone and dihydrotestosterone, are 19 carbons. You can't go backwards. Once you make those, you're destined to only be that. You can't go back to 21 carbons. And then finally, we can take the 19 carbon androgens, lose one more carbon in the form of a methyl group this time. So we just lose a methyl group. We end up with an aromatic ring on the end. The name of the enzyme is called aromatase. And we get our estradiols, right, which have 18 carbons. So we can only go from cholesterol at 27 to progestins at 21, over to mineralocorticoids still at 21, just some oxidation reduction going on. We can take the 21s and turn them into 19 androgens, and the 19s can only be turned into estrogens with 18. You can't go backwards. So I don't need you to memorize all these structures or enzyme names. You're obviously going to have access to this chart. But I want you to know and be familiar with what's happening between the groups. Right? So for progestins to mineralocorticoids, it's redox reactions. From progestins to androgens, we're cutting off that formal group, right? The 2-carbon group at the top. And then from androgens to estrogens, we're removing a methyl group and making the aromatic ring. Okay, so any questions on anything we talked about?